Excellent. So, shalom everyone, and thank you for joining our webinar. Please mute yourself and write your questions in the chat. I will start with a short introduction, followed by a 45-minute lecture by Professor Goda, followed by a 15 minutes Q&A. The webinar today will revolve around a new tool that can help fermentation researchers develop their own strains of microorganisms optimized for the alternative protein industry. We are joined today by Professor Goda, who will talk about his invention on high-throughput Raman flow cytometry. Professor Goda, thank you for joining us and at this late hour. Professor Goda is a professor at several leading universities, both also at the University of Tokyo, University of California, and Wuhan University. His lab recently published on the matter in Nature Communication. So about us, I'm Tom Ben-Arie, a senior scientist at the Good Food Institute. GFI is an international science-based non-profit organization that advances the field of alternative proteins with three main departments. Our SciTech department offers alternative protein courses, counseling, scientific reports, conferences, and databases to accelerate alternative protein research. Our policy department works with governments in order to secure funding and develop favorable policy for the alternative protein ecosystem. And our industry department helps startups and counsel corporations on the field of alternative proteins. We provide industry reports and have company databases and investor databases. Very briefly about the GFI. GFI is a philanthropy-based organization that advances alternative protein as a possible solution for the leading global crises related to animal agriculture, as we all know. If you don't know, you probably wouldn't have been here. And for some reason, okay. And the reason that we need this new technology, as you can see in the this very simple illustration here, is that we need to develop novel raw materials for the alternative protein industry. And one of the raw materials for this industry is microorganisms. And we basically need better microorganisms. And for that, we need to do directed evolution. For directed evolution, the main current bottleneck in research for directed evolution is a selection tool that allows us to choose or sort cells based on parameters that are relevant for our industry. For example, in this illustration, this is a regular fax image. However, our hope is that with Raman flow cytometry, or as you will see soon, that's the case, we can sort cells by their concentrations of protein, iron, and so on. Those are parameters that are important for our industry. So now I will give the floor to Professor Goda. Thank you again for joining us at this hour, and I hope that everyone will find this lecture interesting.
Okay, thank you very much for the introduction. So let me share my screen. Okay. You can see, right? Okay. Well, thank you very much again. So it's 11 p.m. here in Japan, and thank you for coming to my talk over the World Cup game. Actually, I had to go back and watch the game continuously. So my main affiliation is to the University of Tokyo. I also have two adjunct positions, UCLA and the famous Wuhan University, for a negative reason. So my group has been working on the development of the so-called serendipity enabling technologies to realize Louis Pasteur's famous quote, "The chance favors the prepared mind." These are some of my group's representative publications in the past 10 years or so. And today I'll be talking about this Raman image activated cell sorting and kind of related papers. So I assume you have a sort of interdisciplinary background or kind of mixture of people with many different backgrounds. So let me sort of give you an overview of this flow cytometry and cell sorting. And cell sorting of flow cytometry or FACS, fluorescence activated cell sorting, has been around for many years. I think it's been around for the last 50 years or so. Basically, the technology counts, analyzes, and sorts cell populations with high throughput based on scattering and fluorescence measurements. You basically start with a cell population here, and you mix it with the fluorescently labeled antibodies, and you flow those cells into a tube, and then you excite these cells with a laser, and then you measure scattering or fluorescence from each cell, and then you use that information to activate the cell sorter to sort cells one by one. And this technology has been around for many years, and it's been used for many applications in hematology, cancer detection, and microbiology. I think this your audience is probably the most interested in this microbiology applications. While the FACS is quite useful, it has several disadvantages. So cells are interrogated via fluorescence probes in FACS or conventional flow cytometry, meaning that the major advantage is the rich chemical information and high sensitivity. But it comes with several disadvantages, such as cell toxicity, interference with natural biological function, non-specific binding. It's also difficult to detect small molecules. And also, for your audience, I think it is not useful for application in which fluorescence probes are not available. And this is true in many microbiology applications. So how about using Raman scattering or Raman spectroscopy for flow cytometry? So the Raman spectroscopy is based on the measurements of the vibrational modes in the molecule. So you start with the incident light here. The light is incident onto a vibrating molecule here. And then in this case, it's a CO2. And then 1 out of 10 to the 6 photon gets Raman scattered with a frequency shift caused by the vibrational frequency of the molecule. So you measure the Raman scattering signal from the molecule, and by measuring it, and you can identify the frequency shift. And this was discovered by C.V. Raman, and he received the Nobel Prize in physics for the discovery of Raman scattering. And so by measuring this, you can actually identify the species of the molecule without doing anything, without the need for labeling. And so, it was discovered in 1930, so it's been almost 90 years. And it's been used for analyzing cells directly because cells are composed of biomolecules. So this is, I'm not sure, this is a kind of cell here. So the cell contains many molecules, and each peak corresponds to molecular vibrations. Yeah? The x-axis here is a Raman shift. Raman shift means basically the light frequency shift caused by the vibrating molecules. So it has advantages such as the following: no need for tagging or labeling, meaning that there's no cellular toxicity, no interference with cellular function, no photobleaching, no non-specific binding, and also it is possible to detect small molecules such as metabolites. However, there's a clear technical challenge, which is basically a weak signal. As I said, only 1 out of 10 to the 6 photon gets Raman scattered by the incident light or molecule. So improving this low sensitivity problem is the major goal of Raman spectroscopy. So let me talk about Raman flow cytometry and cell sorting. So Raman flow cytometry and cell sorting actually has been around for the last 2 decades or so, because as soon as the facts of fluorescence-based flow cytometry came about, people realized the limitation of fluorescent labeling. So some people started to investigate the possibility of using Raman scattering or Raman spectroscopy for probing cells directly over fluorescence detection. And here I show the figure merit. The x-axis is the bandwidth, because the more bandwidth we have, the more information you get about cells. And y-axis is throughput. EPS means events per second or cells per second. So you want to go that way to the top right corner, right? More information and more throughput. And let me talk about this work here. So this report, so we have recently demonstrated this set of specifications to conquer this unknown area. So it's a little sort of complicated system, but let me sort of go over each subsystem carefully so that you can have a sort of good understanding of what's going on. So cells are flowing here into this microfluidic channel. And then here, cells are interrogated by our Fourier transform coherent anti-stock Raman scattering spectrometer. It's a kind of advanced coherent Raman spectroscopy system, because we are kind of photonic scientists. We developed this kind of high speed and high sensitive Raman spectroscopy system. And then Raman signal is detected here. And then the detected signal is sent to the real-time spectral processor, where each spectrum is analyzed. And then on the processor, we make a decision whether we sort or unsort the target cell. And then decision making signal is sent to the piezo driver. And this piezo driver activates this set of piezoelectric actuators. And this part is composed of 2 reservoirs of liquids. And when the target cell comes, we want to kick the cell out of the mainstream. So we push this membrane and we pull this membrane so that there's a secondary flow in this direction, perpendicular to the main flow, to kick the cell, so that the target cells are kicked into the sort channel, whereas the unwanted cells go straight into this unsort channel. Right? That's how it works. And it's a little detail, but let me sort of talk about. Okay, animation doesn't work for some. These are kind of details of our Raman-activated cell sorting system. It's kind of complicated. But the heart is this part, this on-chip cell sorter. The light comes into this sorter, and Raman signal is measured. And measured Raman signal is sent to this spectral analysis and decision-making module. And then it activates the cell sorter. And everything is controlled by feedback control system. And this is a kind of basic demonstration of bead sorting by Rux. So here we have a mixture of PMMA and PS polymer beads, and they have different Raman signatures. So when the PS or PMMA bead comes, and they have characteristic signatures here, and here when we have unwanted particle, we recognize it as the waste and say the waste. And we have a target particle coming in, and then it activates the cell sorter, and then there's a secondary flow from right to left or left to right. And this is PMMA, right? And this is a PS, polystyrene. And then keeps going on. And as you can see, this is happening at very high speed. And next, we apply this RAX system, Raman-activated cell sorter, to various types of cells, Euglena gracilis cells, chromochloride cells, and hematococcus cells. And each microorganism species has unique applications. For example, like Euglena gracilis cells produces palamylon that has been used for like biofuel, bioplastic applications, and also food supplement. And chromochloride produces starch, and it can be used for many applications such as medicine and food supplements as well. And also hematococcus. Hematococcus is known to produce astaxanthin, which is a carotenoid. And also it is quite useful as an antioxidant. And it's being used as also a food supplement as well. And we actually use the RAX system to see the Raman signatures from each cell. As you can see, that each single cell's Raman signature. As you can see, the Raman peaks are located at the different wave numbers of Raman shifts. And we can use that information to activate the cell sorter. And this the mixture of sorting, sorry, mixture of cells. I think it's a hematococcus and Euglena. And as you can see here, we are looking at this 200 and 472 inverse centimeter Raman shift. And when the target cell comes, I think this is Euglena, or maybe hematococcus, and it activates the cell sorter and kicks the cell out of the main flow. Okay. So this is a kind of summary of the demonstrations. So Euglena gracilis cells, chromochloride, and hematococcus. So we have in each application. So here in the Euglena gracilis application, we started with a low palamylon and palamylon content cells, and then we only sorted high palamylon cells out of the mixture with a throughput of roughly 50 events per second or 50 cells per second with a high purity of 93%. And we have also achieved similar demonstrations like high starch, low starch, and so on and so forth. And these two pieces of work have been published in these two papers. And the next is Raman imaging flow cytometry. So far, we have done only single point Raman measurement based flow cytometry and cell sorting. So now we want to extend the capability to imaging, because imaging actually provides a lot more information about cells. And it's kind of complicated, so let me sort of briefly mention it and maybe skip it over. So we start with a pulse train that activates the molecules in the cells. The cells are flowing in this microchannel again. And then we use stimulated Raman scattering for coherent Raman measurements. And here the cells are flowing and flowing through the center of the microchannel by acoustic waves. It's kind of complicated as well. So, we start with the pulse train and separate it into 4 channels, and then recombine with the design line so that we create this pulse pair resolved wavelength switchable pulses that are used to activate the stimulated Raman scattering measurements in the cells. So these are kind of basic demonstration of the Raman imaging flow cytometry, again using 3 kinds of polymer beads, PE, PMMA, and PSBs. And we use 4 different Raman shifts or wave numbers to acquire Raman images. And these are raw images, and based on the raw images, we developed the decomposed images at the different Raman shifts or Raman colors. So here we can see polystyrene beads, PMMA beads, PE, and Marge. And based on this, we can have a library of stimulated Raman scattering images of polymer beads like this for large-scale statistical analysis. And then we apply this Raman imaging flow cytometry system to Euglena gracile cells. Again, we've been using Euglena gracile cells for microbiology applications and also for studying biofuel production. So here, under nitrogen sufficient condition, we see Euglena gracile cells, and these are flowing at 2 centimeters per second. And when the nitrogen deficiency is applied, which is a common method for inducing the production of paramyron lipids, we can start to see, lipids and paramyron production in the cells. As you can see, the red corresponds to paramyron, and blue corresponds to lipids. Again, without a need for labeling. We don't do any labeling here. And because we can acquire lots of images with a high throughput of 100 events per second or 100 cells per second, we can acquire roughly 10,000 or maybe more images within 100 or 200 seconds per culture condition. Then with the ability to acquire lots of images, we can apply big data analysis methods such as convolutional neural network or CNN to do a more sophisticated analysis of the images. And here we have the t-SNE plot to segment them and then separate these populations. And then this computer matrix of the cultures indicate that this deep learning or CNN classifies them with excellent classification accuracy of 99%. This is probably not related to the Good Food Institute, but it's a liquid biopsy, label-free liquid biopsy application, cancer application. 90% of the cancer deaths is caused by circulating tumor cells, CTCs. And detection of CTCs has been quite difficult because the surface expression or surface antigen of CTC changes depending on the cancer stage. So people wanted a label-free CTC detection schemes to identify CTCs more accurately. So we have applied this Raman flow cytometer to CTC detection. And here the images of the whole blood cells, PBMC, Zika cells, and cancer cells. And again, by just like the Euglena gracile cell application, we applied the CNN convolutional neural network to these cells to develop the confidence matrix, and we have a decent classification accuracy. And then we can use a system to identify CTCs out of a mixture of the CTC and other type of blood cells. So here, the pink color corresponds to lipids, and lipid is a good indicator of cancer cells. And these 2 pieces of work have been published in these 2 papers. And finally, I think this is the main topic. So then based on the Raman imaging flow cytometry technology, we extended that technology to cell sorting. So now we use Raman images to activate the cell sorter to sort cells based on the Raman images. So here's a schematic of the system. So this is a microfluidic chip and cells are injected into this sample tube here. And then cells are focused into a single stream because cells are usually randomized. So they are forced into a single stream so that we can do imaging of each cell one by one. And then here, cells are flowing at roughly, I think, a few centimeters per second. And then those pulses are injected into the cells for Raman images, coherent Raman images. And the Raman images are analyzed on the real-time Raman image processor, where those images are constructed and analyzed. And then decision-making signal is generated and sent to this dual membrane push-pull cell sorter to pick cells one by one. And this part corresponds to the recognition of human brain. This part corresponds to decision-making of the human brain. And this part corresponds to commanding. And this part corresponds to actuation. So in a sense, this is quite analogous to a microscope with a cell picker or a pipetting. But it's done at much higher throughput, 100 times, 1,000 times more efficient than the human actuation. This is a kind of recap. So again, the microfluidic chip is composed of 3 layers. This hydrodynamic focusing part to create a single stream. And then this part is composed of 2 glass membranes with piezoelectric actuators to kick cells. And the more detailed picture is here. Again, it's made of 2 reservoirs. And when the target cell comes, we push this membrane and we pull this membrane so that there's a secondary flow in this direction to kick the cell out of the mainstream. And we can do this in the other direction as well. And this is the brain part. Again, this is a microfluidic chip. Information comes in, and then it's sent to the brain part, which is composed of lots of FPGAs, GPUs, and CPUs to analyze each cell image. And then analysis is done on the brain part, and the decision-making signal is sent to the module. So this is a kind of the video that shows how this work is actually being done. Let me start over. So this is the optical system. It's a kind of huge system. Normally in the morning, we discuss what kind of experiments to perform. And then this guy sets a solution that contains cells into the system. And then we push the start button and monitor each subsystem. And we wait for, like 20 minutes, 30 minutes, depending on the application. And after the experiment is done, we collect 2 tubes that contain sorted and unsorted cells. And then we double check the sorted cells under an optical microscope, check the sorting yield and purity, and also make sure that the collected cells are the right ones. And then finally, we discuss what to do with the sorted cells. Some people want to cultivate those cells or do a directed evolution of these cells, or some people want to do DNA or RNA sequencing of these cells, or maybe more detailed analysis by electron microscopy. And here's a sort of basic demonstration. Again, we always calibrate the system with polymer beads. Yeah, the PSBs and PMMA beads. And this output, the collection outlet and the waste outlet, indicates high purity of sorting. And this kind of shows the images of the cells we sorted. So microalgal cells, the chlorella, Chlamydomonas, Gloeromonas, and also mammalian cells, 3D three L1 cells, the kind of adipocyte-like cells with a lot of lipids, and also microalgal cells with isotopes, and those stem cells. People want to do Raman cell sorting of stem cells because they do not want to stain cells before injection into the human body. So as you can see, we get Raman images of these cells. Different colors like starch color, chlorophyll color, and so on and so forth. And we did some basic applications, cell sorting applications. Here, we did the sorting of adipocyte-like cells for studying obesity. So we initially started with a treatment for inducing differentiation for lipid induction. And then we sorted the cells with very high lipid content from like 12,000 or 11,000 events, and only like 1% of the events or cells was sorted. And second application was sorting of microalgal cells toward cost-effective biofuel production. And we did random mutagenesis. And then we investigated the lipid content of each cell, and we only collected less than 1%, like 0.1% of all the events, all the cells that contains very high lipid content. And this work was published in this Nature Communications. So it's nearly time. So let me summarize. So Raman flow cytometry and cell sorting with a high throughput of roughly 100 cells per second, 100 events per second has been demonstrated. And Raman flow cytometry and cell sorting is advantageous over fluorescence-based flow cytometry and cell sorting in that it does not cause cell toxicity, interference with natural biological functions, and non-specific binding. It is also useful for particularly useful for microbiology application in which fluorescent probes are not available. Fluorescent probes are mainly available for mammalian cells because the money comes from the medicine. But many microbiologists actually do not have that kind of funding, and also the diversity of microalgal species is a lot more. So it's not possible to wait for fluorescent probes. So they want to directly probe the microalgal cells via Raman scattering. So this is where Raman flow cytometry and cell sorting is quite powerful. However, for practical use, more work is needed to improve the sensitivity of Raman flow cytometry and cell sorting. Still, by introducing the coherent detection schemes into Raman flow cytometry, the sensitivity of Raman flow cytometry has been improved significantly, but still it's like roughly 10 times or maybe 100 times less than fluorescence-based measurement. So for more practical applications, more inventions and more new techniques to improve the signal-to-noise ratio are needed. And all these pieces of work have been sort of summarized and reviewed in this review paper. So if you're interested in further reading, please go to this paper and go to this journal and read this paper titled High-Throughput Raman flow cytometry and beyond. And I'd like to acknowledge my group members and the funding agencies. Thank you very much. Thank you very much for listening.
Thank you so much, Professor Goda, for this really interesting lecture. We will distribute the recording for the people who registered for the webinar, so all of you will have a copy. We see one question in the chat, and I will ask another one afterwards. Oh, we see another one. So Elijah asked, is there a range of particle concentrations that work best with RACs?
Okay. I'm also looking at the,
Oh,
the
okay.
chat. Okay, so it's a range of particle concentration that works best with Lux. It depends on the size and you know, so well, size and also many other conditions of the particles. So there's a range of particle concentration, cell concentration that works for Raman-activated cells. So normally we don't want high concentration because not for Raman spectroscopy measurement, but actually for microfluidics. The microfluidic chips are kind of vulnerable to clogging. If the concentration is too high, you know, they sort of rush into the small microchannel and leads to clogging. And so that disturbs the sort of nice flow of cells. So we want to reduce the cell concentration, but of course, if we reduce the concentration too much, we sacrifice throughput. There's sort of there's optimum concentration that has to be determined by kind of trial and error, and also depends on the size of the cells. Okay, should I go to the next question?
Yes.
Go which one? Blah blah blah. Has your
So
team
the next one, go ahead. Sorry.
Okay, has your team attempted to control the position of the laser point to sample specific regions of the cell particles and such as like a membrane, phased nuclear? Yes and no. Yeah, maybe. Imaging is better. So we, instead of doing a single point measurement within the cell, it is actually easier to acquire Raman images so that we actually had a bigger picture of the, you know, the molecular distribution within the cell. And then we don't have to worry about the fine tuning the position of the laser within the cell. Because the cell position keeps sort of shifting because we use hydrodynamic focusing to create a single stream, but still within that stream, the cell position sort of fluctuates because the fluidics is quite unstable. So it's not sort of practical to control the cell position that precisely. So it's actually easier to acquire the Raman images, and then we look at the bigger picture. That's much easier. Okay, next question. What is the availability of such machine? Is a university company want to explain? It's not commercialized yet. It's still at the university level. Again, for commercialization, I still think that more work is needed to improve the sensitivity so that it can beat the FACS and other fluorescence-based measurements. You know, the market of this flow cytometry is pretty much in the field of medicine. So microbiology or food industry is still small compared to medicine in the field of cell sorting and flow cytometry. So if I think I don't know, more interest is well, increasing in flow cytometry and cell sorting without labeling, then I think the company will be interested in commercializing it. So still the technology is still at the university level. Okay. And are you planning to manufacture the device? How can I use the method at my university? My group is not the company. I have startup, but it's quite complicated and it's quite expensive to build such a system. So we have one system no, a few systems available at my university. So if you're willing to come to my lab and learn how to use it or how to develop it and bring the expertise back to your university and rebuild it, or you can bring your samples here and test it here. Either way, now, I'm open to collaboration. So please send me emails if you're interested. Next, could you talk about cell shape and if something like budding yeast may have an irregular shape and yes and no. Budding yeast cells are not so big. Actually, in this talk, I don't talk about yeast cell, but I actually do fluorescence-based image-activated cell sorting as well. And we actually use budding yeast for cell sorting. And actually, the yeast is quite small, like only 5 to 7, maybe 10 micron at most. So it has irregular shape, like a daughter and mother, but still it's okay. And the stability of the flow is quite good. It's not made unstable by irregular shape of yeast. So it's okay. We can still do a lot of nice imaging of yeast in flow. The next, how is the fluorescence effect observed in Raman spectral microbiology corrected for using the fluorescence effect observed in Raman spectra? For us, so in our scheme, in our Raman measurements, we use a time domain technique. So by time gating, we can actually exclude fluorescence measurements from Raman measurements. As we have probably guessed, the fluorescence measurements are fluorescence signals are much stronger than Raman signals. So by time gating those signals, we can actually extract the Raman signals, and so that we are not contaminated by the strong Raman background signals, strong fluorescence background signals. Will it be possible to distinguish between genetically modified cells, assuming you add a few constructs to a cell, and will you be able to detect a difference? We haven't done it yet, but in principle, we should be able to, as long as we have clear Raman signatures that can be distinguished between genetically modified cells and ungenetically modified cells. So genetic modification had to be translated into some kind of animal protein concentration of protein localization within the cells, and if the expression is clear, and we should be able to use Raman images to differentiate those cells. Okay.
I will add, and I will say the last question is interesting. Perhaps in some way you can remove some kind of those genes that for the resistance for antibiotics. I don't know if that's possible, but perhaps that could be interesting. I will ask another question. Sure. Do you think that perhaps AI can be used to perhaps to detect to get a better signal to detect specific molecules more accurately?
Yes, we actually have done it already, and not based on this Raman image activated cell sorting machine, but actually fluorescence image activated cell sorting system we have developed. We actually apply the deep learning directly to the image processor online, so that we can actually use the deep learning to analyze cells in real time and also sort cells. So as you said, yes, the deep learning is sensitive to tiny morphological features that the human eyes cannot detect. So it actually provides a lot finer specifications. Okay, another question. To what extent are the differences technical environment environmental, and to what extent genetics? Do you see progress in directed evolution? So we haven't done the directed evolution application yet. Actually, we are currently doing it. So yeah, please wait for my next papers. That will answer the question. Yeah, we're writing a paper, and we're actually doing it based on yeast. We are using yeast for directed evolution with racks, and we have acquired some interesting results. And we're writing a paper, so please wait for several months.
Professor Goda, thank you so much for this very, very interesting lecture. Oh, I see someone is joining. Perhaps he will watch us later the recording. Thank you so much for joining us, especially at this late hour. I hope lots of luck with your research. Very interesting. And I hope that everyone thought this was interesting as well. And thank you for joining the webinar. And I hope...
Thank you very much.
Thank you.
Thank you.
Have a great day, everyone.
Thank you.
Thank you very much.
Bye-bye.
Bye.